One of the most exciting aspects of cancer research today is the availability of drugs that hit mutant proteins. And we've seen many spectacular successes where these drugs have been introduced into the clinic. But even in these successful cases, we see a very wide range of responses. In addition, there are a number of drugs that have been brought into the clinic targeting particular mutant proteins that just haven't worked for some reason as well as we expected, or only worked in a small number of cases. To understand why these drugs work in some patients and not in others, we need to analyze the entire genome sequence of the tumors. This is a tremendous amount of information and a big technical challenge at many different levels. Most of these drugs are being tested at multiple institutions. What we need to do is to compile all of this mountain of information about clinical responses and genomic information so we can really make predictions about which patients will respond most effectively to which drug. One example of this kind of approach was published recently, in which a failed clinical study, a phase 2 study, had been wrapped up with really only one clear responder to a drug that is known to hit components of the PI3 kinase pathway. But in the one patient that did respond dramatically, she was still alive 2 years after the study and prompted a group at Sloan Kettering to go in and analyze the full sequence of the tumor of the patient who responded so effectively. And when they did that, they identified a mutation in a gene called TSC1, which is a key component of the PI3 kinase pathway. They followed up with more data from more patients who had responded partially, and indeed all the responders had some kind of mutation in the TSC1 gene. Now we think that patients with TSC1 mutations in other kinds of cancer may also respond to this kind of drug in a way that would not have been predicted previously. But to do these studies and to develop the hypotheses, we need to have access to information gathered at multiple clinical sites across many years using different kinds of platform, and have all this information integrated into a way that we can really make sense of and thus improve the outcome of our patients and improve the development of next generation of drugs.